Dear students, good morning to all of you. You are welcome in this class. Today we will continue movement of nutrients in soil and their uptake by plant roots. So first part we have completed movement of nutrients in soil, and then we started the uptake of nutrients or ions by plants or plant root. We also know that nutrients or ions can be taken up by crop in foliar forms also. The leaves can also absorb, the stem can also absorb nutrients. So we started with movement of nutrients from soil to roots. There are three mechanisms: root interception, mass flow, and diffusion. We discussed root interception. We discussed this mass flow and diffusion also. And you can see mass flow and diffusion are important means of transporting ions from soil solution to the plant roots. So we have seen an overview of the ion absorption. Transport systems we have seen in the plant, xylem, phloem are important. You have seen these pictures before. And xylem is very, very important for transport of nutrients from root to the shoot part. And you have also seen how the ions or water will move from soil to the root through root here, then passes the radial. This is radial movement of water or nutrients, you can see, and then reaching the xylem. From xylem, they go upward. And this picture was also shown you, ion absorption by plant transport system. And you can see the root, the cross section of longitudinal section of the roots and different parts are shown. So here you can see that nutrients are taken up by the all parts of the root, whether it is zone of maturation, root hair, zone of elongation, root cap. It depends upon the kind of nutrient also, in which part of the root you will have more absorption of that particular nutrient or ion. So for this, I have also shown you this data that which nutrient is taken up by which part primarily. So for example, potassium, nitrate, ammonium, and phosphate, they are taken by entire root surface. And you have seen the nutrient uptake by the plant roots. We started this topic. And we have discussed the adsorption means coming to the near to the root. Absorption means entry into the plant system or plant roots, and then accumulation in the plant. So there are three steps in ion absorption. Adsorption, they have to come near to the root. Absorption, they are ingested, they are taken inside, and then they are accumulated. After accumulation, they will move through the move radially to the xylem. So absorption. So already you have seen this picture is very, very important to understand most of the things. So I repeat some information about this picture. You can see this is the cross section of the root and root hair. Suppose root hair is there. There are two ways through which the nutrients move from root hair or from the root external part of the root to the inside of the root. So you can see apoplastic path and symplastic path. In apoplastic path, you can see the ions or nutrients will enter the intercellular spaces between cell membrane and cell wall. So they move, and lot of free spaces are there. So you can see they move red arrow, you can see the red one, and it passes through this apoplastic path. It passes through apoplastic path, and then you can see some is also entered into symplas here and there. Some is also it has lateral entry also into the cell, but most of it through apoplastic movement is going through the intercellular spaces. And then finally you see this is a cortex. The all is cortex cells. These are the cortex cells, and then comes the endodermis. endodermis cell, it stops the all kind of movement. But here some selective movement can happen, and it is helped by Casparian strip. This red one line kind of thing. So this is Casparian strip, and which is helping apoplastic movement. So it is helping apoplastic movement going into the symplas. So here at this point, you can see from 3 to 4. So here up to 3, it was apoplastic movement, you can see. And then with the help of this Casparian strip, it can enter into symplas. Symplas means it can enter into the cell of the endodermis. It can enter into the cell of the endodermis, and then from endodermis cell, it can pass on to the xylem vessel. So this is your apoplastic movement. And here it is not that much easy. Lot of things are involved here. Somewhere energy may be involved, somewhere energy may not involve. Maybe passive movement, maybe active movement. Then comes your symplastic path. Symplastic path is it has to cross the cell wall and then interspaces and then cell membrane also. Cell membrane is plasma lemma. So again and again it has to cross the same kind of barriers. And between two cells, you have plasmodesma or plasmodesmata, which allows the transfer of substances, transfer of ions from one cell to another cell. So you can see the black line, this is symplastic pathway. So here you see the ions or nutrients are passing from cell to cell with the help of plasmodesmata. And you can see this is passing, this is passing, this is passing. And finally, it is also coming to the endodermis. Here, from cortex cells, it will go to the endodermis and it will pass again with the help of this Casparian strip. It can pass this endodermis and it will reach the xylem. And in xylem, it is physical movement, physical upward movement, and it can be distributed to the plant parts or shoot parts, leaves, branches, stem, etc. So broader is that from apoplast of cortex, an ion may either be transported across the plasma membrane of a cortical cell, thus entering the symplas, or diffuse radially all the way to the endodermis via apoplast. Just now you have seen. Apoplast forms a continuous phase from the root surface through the cortex. So it is continuous phase you have seen. However, in all cases, ions must enter the symplas. Finally, whether it is before reaching the xylem, they must pass symplas. Whether it was apoplast movement or symplas, finally both have to be symplas before they can enter the stel. Stel is because you have seen that stel is central part of the root, which have xylem, phloem, and some free spaces, hollow tube, because of the presence of Casparian strip. So Casparian strip helps to transfer the material from endodermis to xylem. What is stel? Central part of root or stem containing the pericycle and vascular bundle, xylem and phloem. The Casparian strip is a suberized layer that forms rings around cells of the specialized endodermis. So it is in the endodermis and effectively blocks the entry of water and solutes into the stel via the apoplast. So it helps. Once an ion has entered the stel through the symplastic connections across the endodermis, it continues to diffuse from cell to cell into the xylem. Once ions have been taken up into the symplas, now see the symplas of the root at the epidermis or cortex, they must be loaded into the trachea. Means they must reach the xylem, elements of the stel, to be translocated to the shoot. So whatever movement you have, whether symplas or apoplast, the material has to reach the xylem. The presence of the Casparian strip allows the plant to maintain a higher iron concentration in the xylem that then exists in the soil water surrounding the roots. So Casparian strip has variety of role. Once the ions have been taken by xylem, it will not allow them to come back to the cell. So this is also another advantage of this Casparian strip. Now this was just kind of background. Now you see a different mechanism of nutrient uptake and transport in plant cells. Dear student, it is very complicated subject, very complicated subject. It is not very well understood to me also. I tried my best, but lot of confusion are there. We really do not have good books and good writers who can write the things very clearly so that each one of us can understand it well. So I did my best. I put a lot of efforts to learn this subject, but still some confusion is there. Anyway, I try to get the best thing for you. So in this case, you can see mechanism of nutrient uptake and transport in plant cells. So in this case, you can see nutrient uptake by roots. So first part could be the uptake of nutrients by root. Second part is that they must be transported. Transported once they are taken inside the root or root hair, then they must be transported to the xylem. So transport or movement of nutrients or ion into plant cells. How they can enter the cells and from one cell to another cell and so on. So one is coming to the nutrient uptake by roots. Apoplast, you have seen apoplast, transmembrane, and symplast pathway. This is one option. Second is uptake of ions by diffusion and cation exchange. So these two are the part of nutrient uptake. Apoplast, transmembrane, and symplast pathways, they help in the uptake or entry of nutrients. Then uptake of ions, diffusion and contact exchange. So they also facilitate the uptake of ions. Then transport or movement of nutrients and ion into plant cells. So this happens by three different means. One is passive transport, which includes ion exchange theory, Donan effect and equilibrium, and mass flow. Then active transport, ATP mediated ATPase pumps, cytochrome pumps, phospholipids as carriers. So these are three options under active transport where energy is involved. Then membrane transport. Some nutrients are transported with the help of membranes. So it includes three different pathways. One could be channels, other could be pumps, and these pumps can be divided into two: primary active transport and then secondary active transport. By symplast, it is co-transport and antipode counter transport, and then third one is carrier. So this is how this all mechanism of nutrient uptake and transport are classified or grouped into this. Now coming to the first part, which is your apoplast, transmembrane, and symplast pathways. So you see free intercellular spaces of epidermis, and cortical cells are called apoplast. It facilitates movement of ions or water in root and does not need movement through impermeable membrane. So here there is no need to pass through the plasma lemma or plasma membrane. It passes outside the plasma membrane. Between the cell wall and plasma membrane, you have interspaces, and through those spaces, it is apoplastic movement. Casparian strips force water ions in the apoplastic region to cross the endodermis by passing through the plasma membrane. So this Casparian strips help to pass the materials through the endodermis, or from cell to cell, or it facilitates the entry of material from apoplastic part to the symplast, mainly allowing them passing through the plasma membrane. The cellular connections and plasmodesma help in movement of ions from cell to cell in symplastic movement. In symplastic movement, the materials or ions or nutrients are moving from cell to cell, which is through plasmodesma. These are small structure or small orifices or passages through which materials pass from one cell to another cell. You can see it more clearly. First, you see what is plasmodesmata. Singular is plasmodesma. Plasmodesma is singular and plasmodesmata is plural. What is plasmodesmata? It is tubular extensions of plasma membrane. Tubular extensions of plasma membrane, 40 to 50 nanometer diameter, that traverse the cell wall and connect cytoplasm of adjacent cells. So two cells are connected by plasmodesma, which is 40 to 50 nanometer diameter. And cytoplasm form a continuum because both are connected. Both cytoplasm of both or cell to cell are connected. So cytoplasm form a continuum interconnected by it, referred to as symplasm. And you can see intercellular transport of solutes through plasmodesmata is thus called symplastic transport. So if somebody asks me what is symplastic movement or symplastic transport, it is intercellular transport of solutes. Intercellular means between two cells. Intercellular transport of solutes through plasmodesmata is thus called as symplastic movement. In the picture, you can see these are plasmodesma or plasmodesmata, these pores and cytoplasm. This is one cell, this is another cell. In one cell, you can see apoplastic movement. Apoplastic movement is here. Here, 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 here. It goes like this. And symplastic movement is from cell to cell. This could be the vacuole. And this is your cytoplasm. So from cytoplasm, the materials can go through this plasmodesma to the another cell. And this is in continuum. Symplastic pathway. So both can be seen in this picture. Cell wall you can see, cytoplasm you can see, vacuole you can see, apoplast pathway you can see, and symplast pathway you can see. Now uptake of nutrient ions, you can see here. First one was apoplast, transmembrane, and symplast pathway. Now second you see nutrient uptake of ions by diffusion and cation exchange. So nutrients or ions move towards plant root by diffusion or cation exchange. This already covered with you. Diffusion occurs on the basis of the concentration gradient. Cation exchange happens in response to the release of hydrogen from roots, causing a change in soil pH around the roots. When water or ions enter plant root, they move through apoplastic, transmembrane, or symplastic pathway. Afterwards, movement of nutrients occurs via xylem. Means they must reach the xylem. Nutrient uptake differs with the area or section of the roots. So nutrient uptake means the quantity of nutrient that can be taken up per unit time will differ from area or section of the root where the nutrients are being taken up. Transport or movement of nutrients or ions into plant cells. How they will enter the plant cell. You can see here this transport or movement of nutrients, ions into plant cell. How they can make entry into the plant cell. So maybe passive transport, maybe active transport, or maybe membrane transport. So plasma membrane and tonoplast. What is tonoplast?
Membrane of the vacuole.
Yeah?
Membrane of the vacuole.
Good. So plasma membrane, tonoplast is the membrane of the vacuole. Plasma membrane and tonoplast are the major barriers for movement of ion. Means passing through these two membranes is difficult and selectively regulate the movement into and outside cells. So both the membranes are important and they are semi-permeable to the things. The characteristics of these barriers or membranes are they are permeable to oxygen, carbon dioxide, as well as certain compounds, semi-permeable to water, and selectively permeable to inorganic ions and organic compounds. amino acids and sugar. Selective permeable means they will allow passage of certain inorganic or organic compounds, but for certain they will not allow. Nutrient ions can be transported across these barriers active or passively. So these barriers can be crossed actively or passively. Now you see transport or movement of nutrients ion into plant cells, how they can cross the membrane. So one can be the passive transport, other can be active transport, and third one can be membrane transport. Membrane transport. So I've already listed different approaches or tools in each under each category. Like passive, you have ion exchange, Donan effect, mass flow. Active transport, three different ways, and then membrane transport. Now, passive transport, how passive transport happens, occurs along the concentration gradient. Along the concentration gradient means from higher concentration to lower concentration. Nutrients diffuse into the cell. If the concentration of a nutrient ion falls less inside the cell, then they can diffuse. Energy is not required. Nutrient concentration is maintained for enhancing the nutrient absorption. It happens by three mechanisms: ion exchange theory, Donan effect, and equilibrium and mass flow. Ion exchange theory, exchange of cations present on the soil clay and protons present on the root surface. Carbonic acid in the soil dissociates into protons and bicarbonate ions, and these protons exchange with cations present on the root surface. Now, second one is Donan effect and equilibrium. Cell membrane has a negative charge that allows anions to enter it. Plants can accumulate more than about 30 times the cations passively against the concentration gradient to balance the electrochemical potential difference thus generated. The equilibrium defined by Fg Donan. The ratio, what is equilibrium? The ratio of positively charged ions inside to outside will be equal to the negatively charged ions inside to outside. Now, mass flow. This is also passive transport. Due to active transpiration, ions are taken up from root to shoot xylem. This leads to concentration gradient and a water pressure gradient, which drives the mass flow. Means overall, what you can see in the nutrient uptake and transport, it is not by any single means. It is not by any single molecule. It is collectively. It happens collectively through different means. There are a number of ways that nutrients are taken up by the plants and then they move in the roots. So not a single method or way is available. It is all collection of all things. Now, active transport. Active transport, you know, transport means movement. Happens against the concentration gradient. This is opposite to diffusion. Happens against the, not along the concentration. It happens against the concentration gradient. This type of movement involves energy. There are three major mechanisms for active absorption. How the energy will come. So ATP mediated ATPase pumps. Ions are actively absorbed by using energy released by hydrolysis of ATP, for example, hydrogen ion, ATPase pumps. Second is cytochrome pumps. Cytochrome pumps. Whenever a tissue is placed in a salt solution, the associated respiratory increase is called salt respiration, which is mediated by iron-containing membrane proteins called cytochromes. These are proteins, membrane proteins called cytochrome. Protons and electrons are produced due to dehydrogenation reaction in the inner side of the impermeable membrane due to difference in oxygen concentration. The ferric ions on outside the membrane bound cytochrome pumps accept an electron and get reduced. And subsequently, the electron is transferred or carried outside by the cytochrome chain. This cycle repeats after the next anion is taken up by the ferric ion from outside. Concept was proposed by Lundegaard in 1954. Phospholipids as carriers. Diacyl glycerides like lecithins phospholipids as carrier, diacyl glycerides like lecithins helps in this movement. Now, membrane transport. Membrane transport is mediated by specialized protein, which are described below. There are three options. One is channels. These are transmembrane proteins that exist as membrane pores, which selectively permit solutes based on its biochemical and biophysical properties. The entry of ions is determined by the layer of hydration, that is aquaporins. Then pumps. These are transmembrane proteins that transport ions actively by TP hydrolysis. Protons are pumped against electrochemical gradient, which builds up a positive charge due to the protons accumulated. There are two types of active transport that occur, primary and secondary. Primary active transport, energy from ATP hydrolysis is directly used for proton transport against concentration gradient, thereby creating electrochemical potential gradient. Then secondary active transport. Due to the electrochemical potential gradient that is generated as a result of primary active transport, the protons pumped outside against the concentration gradient result in a chemical potential difference of about 10 to 100 times, which is used for absorption of other ions. So there are two forms of secondary active transport. Just 2-3 slides are left. There are two forms of secondary active transport. One is symport, which is known as co-transport, where proton moves along with another anion in the same direction. Antiport, counter-transport, where the energy generated due to accumulation of protons is used for expelling cations, wherein the protons move inside and cations move outside. Now, third one is carrier. In membrane transport, third is your carrier. This concept defines a specific carrier for each ion. For each ion, you will have a specific carrier. Charged ions move through protein components across a membrane and not through the lipid part. The enzyme phosphokinase mediates the formation of an activated carrier, which then accepts the ion to form the carrier ion complex on the outside of the membrane. The carrier ion complex gets energized and moves through the membrane along with the carrier. Enzyme phosphatase changes the conformation of the carrier ion complex and releases the ion in the inner membrane. Last slide. Finally, you can see these are the important conclusions. The availability of all the essential plant nutrients in soil fluctuates depending on many factors. This may result in deficiency or toxicity of nutrients. The nutrients from the soil move toward the roots by following certain mechanism of transport, which include mass flow diffusion and root interception. The nutrients thus reaching the roots are absorbed by plants either actively by spending energy or passively by no involvement of energy. Dear students, it was really half part of this lecture was tough, and most of the things are even not understood to me because I'm not plant physiologist or biochemist. I just tried to expose you, try to simplify this subject because as a student, you must learn it. You have to learn it, and for this, I tried my level best to simplify it, but still there may be difficulties in this. And if you are in MSc, you can take a course in plant physiology where such kind of subjects are covered. Do you have plant physiology as a minor if you are in MSc?
No, sir.
You do not have?
No, sir.
Which minor you have?
Statistics, sir.
Okay. So one is compulsory minor and other is statistics should be compulsory minor, I think. And there may be one optional minor also. Is it in PhD, I think then? So whatever best was possible from my side on this topic, I did it with limited knowledge. If you like to ask question, you can ask me. Hello? So now we can end it and we meet tomorrow at 12:15 and you can open your videos, please, so I can finish taking your attendance does not matter, but thank you very much for sparing your time. And if you have any question, you can ask question also. Anyway, thank you very much. See you tomorrow at 12:25.
Thank you, sir.